Hi guys! In this video we will be looking at biological molecules and food tests. Biological molecules or biomolecules are organic molecules that make up living things and are essential for living things. So in this topic we will look at 4 biomolecules and how we test for their presence in samples. So we will be looking at lipids. Lipids are fats and oils. We'll also be looking at carbohydrates. And in carbohydrates specifically, we'll look at starch. Let's write that down. We'll look at starch and also glucose. So glucose and starch are both carbohydrates. They contain the elements carbon, hydrogen, and oxygen. And lastly, we will also look at proteins, how we test for proteins in samples. So let's write that down, proteins. Okay, so for biomolecules, lipids, starch, glucose, and proteins. Let's start with lipids. Wait, before we start, we are also going to look at why we need to do these food tests. Like why do we need to test for lipids or starch or glucose or proteins, and the practical examples of that in both the medical field and industrial field. So let's start with lipids. Lipids have the following elements: carbon, hydrogen, and oxygen. Let's say you have a sample and you want to know if this sample had lipids or not. So you've already prepared the sample. What you're going to do is you're going to add ethanol to this sample. And you will shake this mixture gently. And after you shake it, you're going to pour the sample mixed with alcohol, you're going to pour it into an equal volume of water. Okay, so I'm going to put that down, equal volume of water. My pen is having issues again. Equal volume of water. Okay, there. And then you will observe for the results, right? So if it forms a milky white emulsion, that means that lipid was present in the sample. Okay. Since I've used a white background, you can't really see it, but it will form milky white emulsion if lipid is present. Let's write that down. Milky white emulsion. So if the milky white emulsion forms, that means that it tested positive for lipids. That means lipids was present. Okay, let's move on to carbohydrates. And I think we'll use more interesting colors here now. So to test for starch, you will add a few drops of iodine solution. Let's do, okay. You're going to add a few drops of iodine solution to a sample, and then you're going to gently shake the mixture. Okay, so I'm going to write these steps down because in your IGCSE exams, sometimes they do ask for this. So you're going to add a few drops of iodine solution, you're going to shake this sample, and if it turns blue-black in color, that means that starch was present. Okay, so the color here is blue-black. So if the mixture turned blue-black after you added the iodine solution and after you shook it, that means that it tested positive for starch. Okay, next, let's move on to glucose. Oops. Okay, there you go, glucose. Okay, for glucose, you do need to remember, it's a bit special, so you would have to remember this. For glucose, let's say you have a sample and you want to test it for glucose. What you're going to do is you're going to add 2 centimeter cube of Benedict's reagent. Let's write that down just in case I'm pronouncing this wrong. You're going to add Benedict reagent. And then what you're going to do is, for glucose, again, special case, you would also need to heat this sample. So what you're going to do is, I'm going to draw a little mini fire there to show that it requires heat. Yes, but you don't heat this directly, okay? You're going to use a water bath for this. So you're going to use a water bath. Yeah. So the boiling water bath, and you put that in. Let's write that down. You needed a water bath. Okay. So you added 2 centimeter cube of Benedict's reagent to your sample, then you heat it for 2 to 3 minutes in this boiling water bath. And if the color changes to brick red or orange, let's see, yeah. If the color changes to brick red or orange, that means that it tests positive for glucose, meaning that glucose was present in this sample. So let's write that down. This is brick red, positive. Yes, if it turns brick red, that means that glucose was present. Okay, interesting. I like the colors here. Okay, let's move on to the last one, proteins. So again, remember carbohydrates had the elements carbon, hydrogen, and oxygen. Now we're moving on to proteins. And for proteins, there is one extra element there which was not present in glucose and in lipids, and that is nitrogen. So proteins have carbon, hydrogen, oxygen, and nitrogen. Okay, so you have a sample and you want to test whether there's protein or not. What you're going to do is you're going to add a few drops of burette reagent. Okay, you're going to put burette reagent in there. Then you're going to gently shake this mixture. So you shake, shake, shake gently, and then you observe the color. So if after the mixing and everything, if the color changes to purple or mauve color, that means that protein is present in this sample. Okay, I hope this is the right color. But yeah, along the shades of purple, if it's purple or mauve color, that means that your sample had protein present in it. So positive for protein. Okay, so why are we studying all this? What's the purpose of us knowing whether starch is present, protein is present? Why go through all this? What's the practical examples for all this? Let's go to it. So I'm going to give you a few examples. The first one is something that you may have experienced by now. Okay, if you're too young, maybe you don't know, but okay, if you've ever been to the doctor for a urine test, then you would know this. Okay, so urine, you will study in the later chapters as well. It's not supposed to have glucose and protein if your kidneys are healthy, right? But what if there is glucose and protein in your urine? How do you test for that? So this is where, this is a practical example of why we need to know all this stuff. So you do you test urine samples for glucose or protein to see whether an individual is healthy or not, because if they're healthy, they won't have either of these present in their urine. But if something is a bit off, then glucose and protein might be present. Another example would be factory waste, for example. Let's say there's starch. People claim that there's starch in the waters. What are you going to do? You're going to take the sample of the water that the factory waste is going to, and you're going to test it for starch. And this one is really important, spinal fluid for glucose. So one of the tests that's done for meningitis, to test for meningitis, is to check for the concentration of glucose in the spinal fluid, because patients who have meningitis apparently have low concentration of glucose. their glucose concentration falls. And lastly, and I feel like this is a very common example, is when you want to test food for diabetic people, for example. You want to know if food has glucose or not, because glucose is not good for diabetic patients or diabetic people. So you test the food for glucose by performing these tests. And that's the practical example of this whole topic right here. And I hope you enjoyed this video. Please like, share, and subscribe. And thank you for watching.